Welcome back, people. This is the second lecture of our Engineering 340, 542. I'm going to have another class where there's a little bit more lecturing at the beginning than will be usual going forward. I know people have different levels of biological background, and so we're going to talk a little bit of biology at the beginning, and then we'll get into more modeling towards the end of the class. I guess I should ask before we get too much into it if people were able to do the homework all right. I realized that there was a pretty open-ended set of thinking problems for this week. And as always, I need to remind you that this class is live streamed and recorded. Archive. So today we're going to be talking about kinds of biological network. There are many kinds of biological network. We're going to review the four main ones that we talked about very briefly last time. That is metabolic networks, signaling networks, gene regulatory networks, and pharmacokinetic networks. We'll talk one or two words about what's called the fundamental dogma of biology, which is that you start with DNA, you go to RNA, messenger RNA, and then proteins and post-translational modification. And for each example, I will try to give just a few words about how they are, what they do, how they're represented, some of the questions biologically you might ask in the context of trying to understand them, and some of the complexity that we're going to ignore in this class. And then we're going to do some very, very simple chemical reaction basics, talk a little bit about reversibility, mass conservation, and stoichiometry. And then starting from that, we'll build a very, very simple simulation, which is more or less the one we did last week. And we'll introduce the concept of a steady state. And then we will do a little bit of computing to do parameter scan. And I know the last homework problem was trying to figure out on your own how to do a parameter scan. So if that was confusing, then today we'll do it together. So this is mainly going to be chapters 1 and 2 in Herbert's textbook. I'll remind you that I know it's challenging to come up with mini project assignments when we're just starting the class. I'd recommend that people try to find some papers, articles about network dynamics. Maybe, I think I asked you to submit 5 or 10 of them for next week that we could look at together to see if they're good material for a project. I also created that document where you could introduce yourself and some ideas to try to assemble some teams. Again, if we're down to one, if we're down to nine students, then teaming may be less essential, but we'll see. I still would encourage people to work in teams for the project. So we talked a little bit last time about different kinds of networks and where they're used. Again, we'll be focusing on biological networks here. Last year, the year before, we focused mainly on infectious disease networks and immune signaling networks. This year, we're going to be talking more about classic signaling and metabolic and gene regulatory networks. Maybe a little bit about pharmacokinetic networks. But the concept of a network, of course, is something that's very general, goes well beyond biology. And the fundamental components of a network, which are in a sense so simple they seem trivial, although when you actually start working with them, they're not really that trivial, which is the concept of a graph composed of nodes, which have states, and then links between nodes that say how the states on a particular node influence the states on another. And we typically have some concept of directionality for that influence, not always, but usually. And so to show that directionality, we use arrows. And so sometimes you'll call these kinds of diagrams arrow diagrams. And there are different kinds of arrows depending on what kinds of processes you're thinking about. We'll often be thinking about chemical transformation and chemical reactions. In the case of pharmacokinetic models, we're talking about mass transport. And in the case of gene regulatory networks and metabolic and signaling networks, you also have a concept of activation and inhibition. And we also introduced the concept of what a model is, and the idea that a model consists of a structure, that would be something like a network diagram, plus a set of parameters, which could be initial conditions, rate constants, and so on. And that together, that concept of model structure and parameters gives us some set of output metrics. And ideally, if we're going to make models that are useful, we need those input parameters and those output metrics to be things that we can measure. If we can't ever, we don't necessarily have to be able to measure them in real life, but if we can't even conceive of measuring them, then our calculation is fundamentally philosophy rather than science. And we also talked a little bit about the difference between dynamics of networks and dynamics on networks. And so the fact that there's a certain arbitrariness to that distinction. In this class, we will be typically working on dynamics on networks. And that tends to be a little bit frustrating, for example, when people want to do evolutionary theory. Because when you do evolutionary theory, you have a lot of serendipity. Things start interacting that didn't interact before. And that means you're creating links in a network that weren't there before. And so that gets you in the domain of dynamics on networks. And the main issue with dealing with dynamics on networks is dynamics of networks. In principle, if I introduce new nodes, then the game is open. I have no limits to what kind of complexity I can build. But if I have a fixed number of nodes, then in principle, I could create a link between every pair of nodes and just set the strength of the interaction to 0. Neural network people do that a lot, all networks. The problem with those typically is that they're bulky to specify because most of the links are 0. So you have big arrays of links with lots of zeros in them. And so there is some advantage to actually deciding what links are going to be there before you start. Something that is sort of interesting and it was been studied quite a bit in the context of biology is the stability of function when there are missing interactions. So if you think about some network that does something functional, and you ask the question, what if there were a connection in this network I didn't know about? And you add that connection, you could ask the question, does adding that connection break the functionality of the network? A lot of times the answer is yes, it does. But there are some claims that the architecture of networks that we find in living organisms tend to be more robust to those kinds of perturbations than you would expect by chance. And then you build a sort of an evolutionary just so story retrospectively to say, well, if the networks weren't robust to perturbations, then we wouldn't be able to survive with them. We're not going to do much in the way of evolution in this class. I find evolution a fascinating topic. It's a difficult topic to deal with mathematically. A lot of study about evolution mathematically talks about optimization. And when you read the principles of evolution, which are very simple, heritable phenotypes and a concept of fitness, variable fitness, fitness sounds like it has something to do with optimization. In fact, the mathematical concept of optimization or physics concept of optimization and what evolution means by fitness are very, very different. If I want to match a particular pattern in a neural network, for example, I could write a cost function that tells me how far apart my result of my neural network is from my target. But in evolution, there is no target. Fitness is how many offspring you have, and not just how many offspring you have, but how many offspring your offspring have. It's basically the logarithm of the number of offspring divided by the logarithm of the number of generations for fitness. And the limit is the number of generations goes large. And that doesn't necessarily correlate with any optimization of anything in particular. Biologically, things can't be too inefficient, and they have to work. And if they're too fragile, they break. So there are some constraints, but those constraints are pretty soft. And a lot of physicists have done a lot of really ugly work misunderstanding evolution by trying to turn it into optimization problem. The flip side of that is that we haven't really talked about the concept of emergence, but evolution is a classic example of emergence. And the kind of model that we're going to do this semester isn't really that good on emergent behavior. The agent-based models we do in the spring are much better at that, although these kinds of models can be a component of that. So coming back to networks that we're going to be talking about this semester, again, very simple, very, very simple idea. We have nodes. Those nodes have states on them, which are represented by values. The links define how the values on one node affect the values on other nodes. And we're going to be putting on each link a rate law, which describes in a mathematical way how that change occurs. And that's, again, this very general formalism that we could use in all sorts of contexts. We talked a little bit about the idea of a chemical reaction. And one of the things that we noticed is that when we convert the chemical reaction to mathematics, the number of lines of math and the number of lines of the chemical reaction aren't quite the same. If I have a chemical reaction, A plus B goes to C at a rate VF of A and B, then that generates 3 ordinary differential equations, a rate for the destruction of A, a rate for the destruction of B, and a rate for the creation of C. And so that one chemical reaction formula gives me then 3 ordinary differential equations. Similarly, we'll find that if A participates in multiple chemical reactions, one ODE for A might actually map to different pieces of multiple chemical reactions. If A plus B went to C and A plus D went to E, then I would have 2 terms in my rate law for dA by dt. And so one of the exercises we'll do next week probably is just do a little bit of mapping back and forth between the arrow diagrams and the ordinary differential equations. It's not difficult, but it does take a little bit of practice to get comfortable with it. Because we're going to be using the arrow diagram notation, the rate reaction notation, and a lot of the publications use the ODEs, it's worth spending an hour or 2 getting familiar with how to go back and forth. The translation is purely mechanical. You don't really need to learn very much, but you need to be able to do that in your head pretty comfortably. And we also talked about the law of mass action a little bit, that if we have A and B reacting to form C, and we think of those as chemical reactants, the one thing we know is that the rate of creation of C better go to 0 when A is 0, and when B go to 0 when B is 0. And the simplest functional form that has that property would be K times A times B. If you're used to dealing with logic, you may remember that an OR is fundamentally addition, and an AND is fundamentally multiplication. So this isn't like an AND, an analog AND. There are other times when we'll have ORs, and actually implementing OR is a little bit more complicated. We began to talk about the syntax of antimony and tellurium, which is what we're going to be using in this class. Again, the main reason we're using antimony and tellurium in this class are two. One of which is the main one that probably is important to you as students, which is that it's very simple. We need to learn about 6 different things. I guess, Joel, how long did it take you to learn antimony? About an hour, right? 2 hours. So it's not hard to learn. It's not as powerful as a language like Mathematica, but it does what we need, and it's very simple. The other reason that it's useful is that and this is probably of less interest to students, but is more important in the practice of science, which is that it's a standard language. And that means that models that you write in antibody are reusable. And they can be converted into a standard called Systems Biology Markup Language, SBML. And SBML is what's called a sharing standard. It's a way of specifying models so that they can be combined, archived, reused. Because rather disappointingly, one can understand perhaps that experimental biology, repeating experiments, is not so easy. But a surprising number of computational biology papers are not repeatable either. And if it's supposed to be science, then you should be able to reproduce the results. And it's amazing how many papers come with 6,000 lines of C++, which doesn't compile, and when you do get it to compile, it doesn't do what they said it did. And so one of the advantages of what we're going to do here is that everything is in plain view, and it's compact, and the data analysis is also pretty compact. And so in principle, you can generate an article where you publish not only all the methods that you use to generate the simulations, but you could also publish all the transformations you made to give the figures. And so those kind of things, it means that it makes the science a little bit more reproducible. It's even more important in a sense as engineers, because if you're going to be doing engineering and biology, you're going to be doing a lot of life critical work. And so it's really important that people be able to reproduce things. And historically, one of the reasons the Environmental Protection Agency came to work with us using Computational years ago was because Computational was open source. And at that time, we were the only open source multi-cell modeling framework that was available. And the EPA had a nice comment, which I guess I should say, it does not represent the opinion of the EPA. It represented the opinion of one person at the EPA who was not representing the EPA as an agency, which was that when the EPA makes a decision, the Environmental Protection Agency makes a decision, if they ban a chemical, they get sued. If they permit a chemical, they get sued. If they don't do anything, they get sued. And so the one thing that they want to do is be able to show that they weren't biased. They can't necessarily avoid making mistakes, but they do want their decision process to be public so that anybody could see it. And so that was really the main reason they came to us, because we spend a lot of time worrying about how to make things accessible. And unfortunately, coding in things like MATLAB or C++ or Mathematica hides a lot of information. It makes it actually much more difficult to reproduce. So sometimes when you run into limitations that you may find frustrating when we're working with antimony and tellurium, you may be, it won't perhaps make you feel less frustrated, but you might remember that some of the reasons for those limitations are precisely to make the things reusable in a particular way. So now we're going to come back to this set of figures that we talked about a little bit last time. which is these three basic kinds of biological network that we're going to be talking about in this class. And I think we're probably going to be talking mostly about signaling and gene regulatory networks, depending on how many additional projects I could work up during the semester. And we have really three different systems which are very tightly connected to each other inside of a cell and between cells. Metabolic networks are the networks that create the energy currency of the cell. They build the components of the cell. They build all the parts of a cell. And they do most of the primary chemical transformation, especially covalent bonding. Signaling networks control the dynamic state of the cell. And gene regulatory networks turn on and off systems within the cell in a more permanent way. And so I guess I'm trying to think of a metaphor for that. So the metabolic network would be your car engine and how the car runs inside under the hood. The signaling network would be your response to the local traffic in the environment. And the gene regulatory network would be more like having the car modified to increase its efficiency by adding a catalytic converter. Actually, it won't increase its efficiency. It will decrease its efficiency, but decrease its pollution. So we do have these three different kinds of network. They're going to be biologically fundamentally different in a variety of ways. And those differences are going to make differences in terms of how, actually the models are going to look rather similar, but they're going to make some differences when we think about how they function. So I've got some questions, and in this beginning of this class, I'm going to try to outline some sort of kind of questions that sort of frame. Again, I don't know how many people in the class come from different backgrounds, but some people I know haven't done a lot of biological background. And so one of the things I think is important is to try to think about what kinds of biological questions one can frame. And so the fundamental question you might say about gene regulatory network is, how does the identity of the active networks in the cell change? When I talked about the difference between dynamics of networks and dynamics on networks, gene regulatory networks really turn on and off networks completely, de novo. And so in a sense, you could think of gene regulatory networks in a sense as turning on and off networks. So they're in a sense dynamics of networks rather than on networks. The signaling networks typically work with what is available in the cell at any given time, given its gene regulatory state. And so they change the state of whatever networks are active at a given time in the cell. And they tend to control cell behaviors in a dynamic way. And then metabolic networks control production of components and energy. And again, we're always interested in how these things respond when we stimulate them, either in a natural way, in the disease state way, or in an engineered controlled way. Time scale is typically important when we have to combine them. Metabolic networks tend to happen pretty fast, microseconds to seconds, depending on what we're talking about. Signaling networks take a little bit longer, seconds to minutes. And gene regulatory networks take minutes to hours, and that's going to come back to the central dogma of biology, we'll get to in a minute. If we really want to talk about metabolic networks, you could take a whole class on metabolic networks. And if you look at a textbook like Paulson comes from a metabolic network background. And they tend to, metabolic networks are complicated. There are many hundreds of thousands of years of research that have gone into understanding the chemical transformations, in particular the energy cycles in cells, and of the synthesis of the components of cells. And this is a small piece of the fundamental glycolysis pathway in the cell. which is basically the energy conversion, the breakdown of glucose to form ATP. And if you're a biochemist, you'll do better than I will. I can't say I've ever taken a course in biochemistry. It's a fascinating topic, but not my particular area. There used to be one of the companies that sells reagents used to have a poster, which was a poster of the energy pathway of the cell. And over the years, it got bigger and bigger and bigger and bigger. And now these things are mapped out in so much detail, it would be impossible to fit it on the walls of all of this room put together. Unlike a lot of gene regulatory networks and signaling networks, a lot of the rate constants and rate equations in these metabolic networks are known. They've been measured in enormous detail by people. And one of those measurements is to be years of work. Nowadays, maybe a little less, but in the old days, years of work, often using things like radioactive tracers. There's certainly a whole world in, for example, rational drug design, trying to understand what happens if you go in and interfere with one piece of one of these cascades. So there's a lot of complexity here. When people really want to model this, typically because these reactions are so fast, they use steady state analysis. They use what's called flux balance analysis. So they actually don't look at the dynamics. They assume things equilibrate very, very fast. We'll come back to that. And then the mathematics they use are a little bit different. So I'd recommend, if you're interested in that, Paulson textbook is quite good. But you'll see in chapter 1 of Paulson's textbook, they'll say, download the 4,000 element reaction network and solve it. So things get pretty complicated pretty fast, mainly because the connectivity is very high. You see, there are lots of cross links and arrows between components that make it complicated. But ultimately, metabolism is about how a cell takes up nutrients, typically glucose, but also other sugars and sometimes other components. It produces a set of common precursors. Cells like to build standard building blocks, just the way engineered systems like standard building blocks. This figure says a small number of common intermediates, precursors, and cofactors. I think that's a little bit optimistic. The number is pretty large, pretty intimidating. But there are still a fairly limited number for things like energy currency, NADH and ATP are almost always the sources of energy. For lipids, there are a limited number and so on. So they're going to be a set of basic components that, again, if you want to take a biochemistry course, they'll go into a lot of that detail. And then what the cell does, and this is how the synthesis works, whether it's a DNA, RNA, or proteins, is that it takes those limited number of components, like the bases, and assembles them in all sorts of different organizations. Now, there's also a lot of post-translational modification that goes on. Lipids aren't proteins. Sugars aren't proteins. And so all of those things involve complexity beyond the fundamental dogma, but those are classic examples. One thing that you run into with metabolic networks, which we won't talk about at all in this class, it happens a little bit with signaling networks as well, is that some of these molecular species can exist in lots of states. And the problem with that is that mathematically, each one of those states is a different equation. And so if you have, say, a quinone molecule is the classic example, which is used in the mitochondria energy synthesis, also in things like photosynthesis. So quinones have electrons that sit in different places on the molecule. You can think of that as, you can think of a quinone as a big string of bits, and whether you have an electron at a given place is a 1 or a 0. And you could have a quinone that has 16 ones or 0 spaces in it. That means there are 2 to the 16th possible configurations of that one molecule. And that means in principle, to describe that one molecule, you need 2 to the 16th rate equations, which you can't deal with. And so there are some what are called rule-based methods that try to deal with that. They deal with it by assuming that it doesn't matter where you put the electrons, as long as there's n electrons, it's the same. Sometimes that's okay, sometimes it's not. But there's a nice language called Bioengineering that's designed for doing that specific problem. But that is what's called a combinatorial catastrophe. And there's really not much one can deal with. I mean, that's just one of the complexities. It's an irreducible complexity in biology. If we're going to do metabolism here in this class, we'll do just little pieces of these networks. We're not going to do the big networks. If somebody wanted to do a project involving one of these, it's fine. I'll warn you that when people have tried this in the past, they get pretty sticky. They're sort of tarball type projects. They can be pretty challenging. But the kinds of questions that we want to ask when we're dealing with metabolic networks, there are plenty of diseases that have metabolic components. We'd like to know, for example, if we change the amount of substrate, what happens to the rate of production of product? That may sound very abstract, but if you ask the question, if I increase the amount of cholesterol in my diet, what happens to my blood vessels? That's fundamentally a metabolic question. And the fact that there is no simple answer to that illustrates one of the complexities of dealing with these problems. By itself, increasing the amount of one dietary component doesn't tell you what's going to happen to outputs. You might say, well, if I increase the amount of cholesterol, what happens to the rate of fat deposition by liver? The answer is, well, most of the time, depending on the cholesterol, you're going to get more fat in the liver, but not always, and it depends on a lot of other things. On the other hand, there are also plenty of situations in which you can modulate metabolism in a variety of ways. In a lot of simple scientific situations, and also in things like bioengineering for bioreactors, where you use, say, bacteria to make a chemical product, those are somewhat simpler systems. And so then you could ask the question, suppose I have a vat full of bacteria and I'm trying to make molecule X, and I want to make molecule X as much as possible at as high a purity as possible. And what temperature do I need to keep my vat of bacteria at? What density? What pH? What amount of glucose or other sugars do I give them? Those are sort of basic bioengineering questions that one might want to ask. How much oxygen do they need? Do they need carbon dioxide as well as oxygen? And one of the things that we do classically, both in bioengineering and in biochemistry and in the science, is what are called knock-in or knock-out experiments. Knockout experiments are usually the easier thing to do. If I get rid of one of these components in this system, what happens? That's changing the structure of the network. Alternatively, you could say you're setting some components to 0. Interesting things can happen in metabolic networks. A lot of times, the pathway from something that you need to something else that you need goes through steps that have components that can be quite toxic if you get too much of them. And so if you interfere with the pathway downstream, somewhere in the middle, you can accumulate toxic intermediate components. That's something that we've worked with in our lab a bit over the years. When you deal with acetaminophen, So Tylenol. Tylenol by itself is mildly toxic. It's not very toxic, but mildly toxic. Certain cells in your liver convert it to a molecule called NAPQ. The Q has quinone in it. We were talking about quinones earlier. And NAPQ has the unfortunate side effect that it binds to a molecule called glutathione that regulates reactive oxygen species in your liver. And if your liver cells have their glutathione level go down more than about 25–30%, they die. And they die in a particularly inflammatory way that causes the liver to fail. And so every 10 or 15 years, the world rediscovers that acetaminophen is extremely toxic. Usually when a fraternity student drinks a lot and takes Tylenol for the headache and dies of liver failure a week or two later. And then there's a Senate hearings, and the Consumer Reports does a cover story on it. Everybody says don't use acetaminophen, and then they forget about it again. But you could say, well, why would this happen? Well, there are two things. One, acetaminophen is not a molecule your body encounters in normal life. It's a synthetic molecule. But two, the breakdown of acetaminophen into NAPQ and then into its elimination from the body is part of a normal process that your liver does to get rid of what are called xenobiotics, molecules that are not things that you need to keep. It's not that NAPQ by itself is a bad thing. It's a question of how much you accumulate. Provided you get rid of it fast enough, it doesn't do too much harm. And so that would be an example of a toxic metabolite. The source molecule isn't too bad. What NAPQ gets turned into isn't too bad, but there's this intermediate. If you build up too much of the intermediate, you have a problem. And so, in fact, if you knock out the molecule that creates NAPQ, then acetaminophen isn't toxic anymore. Of course, then your liver doesn't get rid of certain things that you might need, but that's a different question. So knockouts are a classic experiment. And there's a famous study by Rao where he asked the question in bacteria. I think he was working with E. coli. It's a long time ago now. And he asked the question, if you knock out a single gene, are those genes lethal? And the answer is a lot of them are. You knock them out and the bacteria die. But then he showed that you could rescue the bacteria in a lot of cases by knocking out a second gene. And that initially seems rather counterintuitive. You knock out a piece that's critical, the system fails, you wouldn't think that knocking something else out would fix it. But if the problem is a pathway where you have a critical toxic intermediate, and what you've done is knock out the getting rid of the stuff, if you then don't make it in the first place, you could actually rescue. So that's the kind of thing that you can do in this situation. Well, I probably need to speed up a little bit, but these are topics that I think are of, I hope they're of some interest to people and give you some context for the things we're going to do. Again, if we're writing chemical reactions, we've already talked about arrow diagrams of chemical reactions. Typically, we'll write chemical reactions with unidirectional arrows and rates on them. If we use bidirectional arrows, we'll put a rate constant above it and below it as well. There's a little bit of heterogeneity in the way these arrow diagrams are done. Quite often you won't see the rate law written next to the arrow. I will always try to put the rate law next to the arrow. The chemicals that are used up in a reaction are called substrates. The chemicals that are created are called products. Node states, nodes are chemical species, and states are either amounts or concentration. In this class, typically we're going to define states as concentrations, but there are some fields in which it's more usual to talk about amounts. And the problem of converting from an amount to a concentration is you have to know the volume you're talking about. And that's something that's external to the diagram. The diagram doesn't tell you how big a box you're in. Another thing, and this is something we're going to run into a lot, is the idea of a catalyst. And when we come to talking about signaling in regulatory networks, this is going to be our fundamental concept. So here we have a species S1 that's converted to a species S2. In classic molecular reaction, the rate could only depend on the concentration of S1. But here we have a molecule A for an activator. And when A is present, it changes the rate of conversion of S1 to S2, usually increases the rate of conversion for a given concentration of S1. And the key thing here is that A is not used up in the reaction. It's not that A reacts with S1. A could be, say, a platinum surface, and the reaction is happening on the surface. So the amount of A is important, but it's not used up. And because it's not used up, we need a different arrow to describe it. We can be sloppy about that. And we'll typically use this lollipop arrow to represent something that increases the rate without being used up. And you'll notice there's a fundamental difference. A is pointing not at a species, but at an arrow. So you know immediately that there's something strange if you have a species pointing to an arrow rather than a species pointing to a species. And we'll see this again and again as we go forward. If somebody is sloppy and used a pointed headed arrow, A pointing with a pointed headed arrow to the arrow, at least you'd know that there's something inconsistent, because you shouldn't have an arrow that represents chemical transformation with a pointed head. And if it's pointing at an arrow at a rate, it can't do that. There are too many standards for representing metabolic networks. As I mentioned, metabolic networks tend to have lots of components in and lots of components out. There is a standard called Systems Biology Graphical Notation Standards, which tries to regularize the representation of these networks. It's nice when people use it. There are tools for making beautiful diagrams with it, but it's not universally used. As I say, the great thing about standards is there's so many of them. So that's something, but that's something you might want to check out if you're interested in these kinds of things. Something else that we're going to neglect throughout this course, although you can put compartments into the models we'll build, is that metabolic networks are very often spatially and temporally segregated within cells. In particular, in eukaryotic cells like ours, the transformation of glucose into ATP and NADH is done inside of the mitochondria. Evolutionarily, mitochondria were probably bacteria, archaea, that were either symbiotic or parasites of some other cell type. And they still have their own DNA, which has slightly different genetic code from the rest of your body. And the bacteria have, the mitochondria have some offered properties. They produce a lot of reactive oxygen species, which are fairly toxic to the rest of the cell. They have rather complicated internal structure and also external structure. Historically, mitochondria were usually drawn as little looking like little E. coli. In fact, they tend to be connected to each other. They tend to be sort of multi-connected sausage of mitochondria in the cells. When cells are going to die, one of the main ways they die is by mitochondrial permeabilization. The membrane of the mitochondrion gets holes in it. It releases a lot of components that are necessary for chemical reactions inside of mitochondria, are exceedingly toxic to the cell. And Giuliano and Joel have been doing some modeling related to the way cells die. But most of your energy budget inside of a cell is not in the main cytoplasm of the cell. It's compartmentalized into the mitochondria. There are a lot of other rather mysterious organs inside of cells, in particularly the Golgi apparatus and the endoplasmic reticulum. These are all compartments of the cell that are surrounded by lipid membranes or multi-layered lipid membranes. And you have essentially small volumes of material that are transported within the cell. Exactly how they're transported and targeted is still not 100% understood. And that spatial segregation and temporal segregation is pretty complicated. Again, you often will see simulations which just have a box that says mitochondrium in it, or a box that says Golgi apparatus in it. But in fact, you have to remember there is a rather complicated set of spatial considerations that are going on. During protein synthesis, cell synthesis is done in different places during the process of synthesis. Again, we're going to typically neglect all of that. It's actually rather remarkable that any of these models work, given how much is left out. Perhaps one way to put it is this: there's a huge amount of machinery that's devoted to spatial segregation. And normally, if there's that much machinery devoted to segregating these components and putting them in specific spatial relationships, you'd think that those spatial relationships would be critical. And therefore, if you build a model that neglects those spatial relationships, you might think it wouldn't work very well. So those are some of the challenges that you might want to think about. Another thing that happens is that when molecular species are in vesicles, they get actively transported. The vesicles bind to molecular motors. Those molecular motors attach to the cell cytoskeleton and get moved around. And that's a whole set of additional complexity, which again, we're going to neglect. So those are just some of the thoughts one might have about metabolic networks. Protein signaling networks are the thing that we're probably going to talk about the most during class. These exist fundamentally in relaying information from outside the cell to inside the cell for the cell to make some decisions. They also have to do with how the cell makes decisions based on its internal state. So these are essentially processing and aggregating information to make certain kinds of decisions. In particular, the cells have sensory molecules on their surfaces, receptors. Those receptors could bind to other chemicals in the environment, either free-floating chemicals in the environment or cells that are chemicals that are bound to the surface of adjacent cells. And they tell the cell information about what its environment is. They could be mechanical signals, they could be electrical signals, they could be chemical signals, pressure signals. There is a whole standard set of machinery of a number of basic types that cells use so that when a receptor binds, it will release usually what are called kinases inside the cell. Those kinases are molecules that add phosphate groups to other molecules. And the basic architecture of signaling networks is adding phosphates and taking phosphates off of other molecular species. On the left, you see a picture which is showing two cell membranes, some receptor proteins on the surfaces of the two cells and their interactions and some signaling downstream of that. On the right is another one of these attempts to build standardized language for describing that complexity. There's even less agreement on standardization of the representation of this kind of network than there is of metabolic networks. Modeling software like Cell Designer gives you some tools for building that kind of pictures. So again, when we're talking about signaling networks, typically we're going to assume that the components of the network are there. The amount of each one or the concentration of each one can change in time. Its state can change in time. But whether the molecule is present or absent is not going to be changing during the period we're doing the study of. And typically our inputs will be the molecular concentration. And the output might be a molecular concentration, but very often what we care about is something behavioral. And so now we're going to be mapping concentrations, which we can measure in principle, to something that's more diffuse, like does the cell divide? Does the cell grow? Does the cell die? And having a quantitative relationship there is often challenging to determine. And so another thing that we're going to see classically is that signaling networks may or may not involve transport. There's always going to be a little bit of transport and a little bit of chemical transformation. But it's not going to be the whole scale remodeling of molecular molecules that we see in metabolism. A classic example of this, well, there are two of them in this picture. On the top, we see that yellow molecule, which is delta, is binding to that green molecule called notch. And there's a little picture of the scissors. When delta binds to notch, what we see is that notch then gets cleaved. It's broken in pieces, which is what's happening on the right. And that notch cleaved domain then enters the nucleus, the cytoplasm of the cell and sends a signal. So that's actually a chemical transformation because the cells actually, the notch molecule is actually destroyed in the process. Underneath it is a classic signaling phosphorylation cascade. Kinases are molecules that attach phosphates to other molecules. Phosphatases take them off. And the classic example of a kinase is MAP kinase. MAP, MAP, you'll see K a lot of times for kinase. The notation is not always standardized, but if it has a K in it, it's almost always a kinase. And just remember, kinase is something that attaches a phosphate. And so here we have MAP kinase and MAP kinase with an asterisk on it. MAP kinase has a binding site for a phosphate. If the phosphate's bound, you'd say MAP kinase with an asterisk. And that's reversible. It can go back and forth. What attaches a phosphate to MAP kinase? It's MAP kinase kinase. So MAP kinase kinase takes a phosphate that it has here. MAP kinase kinase attaches a phosphate to MAP kinase. In the process, it dephosphorylates, so it would go back to being MAP kinase with no asterisk. And then on top of that, MAP kinase is regulated by MAP kinase. And so you get these stacks. And you could ask, why on earth not just do it in one step? Why don't I just regulate MAP kinase directly? And the answer is typically that if I want something that turns on very sharply as a function of the concentration of a particular molecule, I need what's called cooperativity. And one way to do it would be to have 5 different phosphates have to bind to the same molecule. Another one is to have one phosphate bind sequentially to 5 different molecules. In both cases, the amount of phosphorylated MAP kinase, the MAP kinase with the asterisk on it, will then be a very sharply changing function, almost a step function, of whatever is upstream of the MAP kinase. And so that's something that we'll see a lot, and Herbert talks about it a little bit in his book, but it's something which will reference, for example, a book by Uriel Lone. We'll talk about that a lot more. Signaling networks, there's often a concept of an activated and inactivated state. Typically, the phosphorylated molecule is the active one because it's going to become active by transferring that phosphate to somebody else. But it's not always true. There are molecules which are active without the phosphate and inactive with the phosphate. Biology loves to break rules. There are general patterns, but there are lots of exceptions, like English and spelling. There's a general relationship between the way it's spelled and the way it's pronounced, but there are lots of exceptions. And so biology has themes, but there always are going to be exceptions. A typical timescale of signaling is in the seconds to minutes, sometimes a little bit slower depending on how much reorganization goes on. So signaling is very critical in understanding almost every biological phenomenon. If I ask the question, if I have cells in a dish or in my body, and there's a growth factor, and we'll see a lot of growth factors in the kinds of models we do, maybe not so much in this class, but if we go on with it, you'll see them. And you say, if I increase the level of growth factor in the environment, does the cell grow and divide, or does it die? Winds up growth factors could do both things. Even the same growth factor on the same cell type could do both things under different contexts. If I increase or decrease the number of receptors on a cell for a particular molecule, does the cell increase or decrease its rate of migration in response to a chemical in the environment? Both of those questions are very important if I want to understand cancer. They're also very important if I want to understand engineered tissues or wound healing. If I mutate a sequence, a genetic sequence of a molecule, does the resulting protein act more efficiently, less efficiently? What happens to the system? That tends to be an almost impossible question to answer, unfortunately. A lot of molecular dynamics is devoted to trying to understand that. And then if there are mutations in the network, because some component of that network is interfered with, does that change the outputs of the cell in a way that leads to disease state? Again, that's classic question of cancer, also degenerative diseases. In cancer, you'll often see cancer described as a genetic disease. And cancer generally involves mutations in cells. But fundamentally, you don't care about a mutation in a molecule unless that molecule changes the way the cell behaves. And so ultimately, diseases like cancer aren't diseases of single cells, they're diseases of systems. And we have to be able to link molecular concentrations to behaviors. And that's actually rather hard to do. Modern molecular biology is extremely good at quantifying genes and proteins and other molecular species, much less good at quantifying cell behavior. Those are the things we care about. Notationally, we still have the idea of a chemical reaction. Here S1 goes to S2. In this case, our A stands for an activator. That could be a signaling molecule. We have an idea of an inhibitor. And so what does the first diagram mean? S1 goes to S2 with A. The lollipop means that the more A we have, the more S2 we get per unit of S1, or the faster the reaction. R means, with a T or a blunt-headed arrow, means that the more R we have, the slower or the less S2 we get per unit of S1. Again, the key thing here is that A and R are not used up in the reaction. And so this idea of activation and inhibition is critical. And this is going to be something that we're going to run into again and again. And so activation and inhibition are the classic behaviors of signaling. I'm not going to do all the math here. We'll come back and have a lecture on trying to translate how to sort of formally translate these diagrams into equations. But it's worth just thinking about how this works. If I have a fixed amount of S1 and A activates the production of S2 from S1, then what we expect is that if I increase the amount of A, I increase the amount of S2, or I increase the rate of production of S2, depending on which I'm thinking about. And so we have a bit of a diagram like that. It's also true that often, even if I don't have any A, I get some S2. We'll see some alternative notation, which is very compressed, like A lollipop S2. And that doesn't really make any sense. What that really means is that something I don't know about is producing S2, and A is regulating the rate of production of S2. Again, this will make a lot more sense later, but you're going to run into it in some of Herbert's discussion and homework in chapter 1. Formally, if we want to convert everything into chemical reactions, here I have a picture. A activates S2. What that really means is that A regulates the production of S2 from S1. If I don't care what S1 is, it's some set of chemicals that the cell just happens to have as substrates, then I can say nothing goes to S2. And that means that nothing goes to S2 as the chemical reaction, and the rate depends on the amount of A. And if nothing is going to S2, then the only thing that rate can depend on is A, because there isn't an S1 that can change the rate. Similarly, for an inhibitory reaction, R inhibits production version of S1 to S2. If I don't care about S1, I have nothing going to S2. And that means that the rate of production of S2 is a function of R. And we'll see that empty set, the symbol used at the end of an arrow to indicate something that is actually there, because we believe that chemical reactions conserve mass, but that we're neglecting in the system. Just the way metabolic networks have complicated spatial relationships, signaling networks have at least some spatial components. In particular, a lot of signal transduction is done at the membrane by transmembrane proteins that take signals across the membrane without letting molecules cross. That allows the cell to know what's going on outside without exposing it to the risk of something getting into the cell. There are plenty of signaling molecules like estrogen that do get across the cell membrane and act directly. At the inside of the membrane, there are special things going on, in particular interaction with the cytoskeleton of the cell. Within the cytoplasm, there is reeling of information. That information then often will either remodel the cytoskeleton or will get to the nucleus where it'll have effects on gene regulatory networks. So that there is, again, a spatial component to signaling that can be quite complex. But typically, signaling happens in basically 4 regimes: outside the cell, at the cell boundary, near the cell, just inside the cell membrane, in the cytoplasm, and then in the nucleus. It's a little bit less structurally complex than metabolism. I will see a series of basic motifs. The most important one of which we've talked about a lot, which is phosphorylation, attaching a phosphate group to a molecule or removing it, phosphorylation with a kinase or dephosphorylation of the phosphatase. Both of those things can happen spontaneously at a low rate, more dephosphorylation. You also have complex formation, which is the association of 2 molecules together, not with a covalent bond, with a hydrogen bond or something else, something that's easier to break. Something else that we get pretty often is that the degradation of molecules can be programmed. A classic degradation is what's called ubiquitination, which is the attaching of a tag to a molecule that directs it to particular organelles that break it down. And so that's essentially labeling something as get rid of this molecule. And so that's active regulation of degradation. There's this comment that proteins can have multiple phosphorylation sites. And exactly what each phosphorylation site does with relation to the others is a problem. And again, if you have a protein which has 20 phosphorylation sites, you have 2 to the 20th combinations. In principle, it means you need 2 to the 20th equations. It's hopeless. So we can, in this class, we're going to just duck that question, but in reality, sometimes it becomes true. Now we're going to move on from there to gene regulatory networks. I'm a little behind, so I should probably speed up a little bit. The fundamental dogma of biology, which I hope everybody knows, if you don't, that's fine, is that you start out with DNA, and DNA is a sequence of bases that specify in 4 letters the genetic code. DNA is transcribed into RNA inside the nucleus by particular motor proteins, by RNA polymerases. That RNA initially has things like introns in it, extra pieces which have to be eliminated. And so that RNA is spliced, also has something called the poly A tail attached to the end of it. That spliced RNA, sometimes the same gene can be spliced in multiple ways, is then directed to the ribosomes, which are some of the more mysterious macromolecular machines inside of cells. The ribosomes read the triplet base code and build a protein sequence by attaching components of the protein one at a time in combination with the to match the sequence on the DNA or the RNA. That protein, that proprotein itself is then released from the ribosome. Your cells have a lot of ribosomes in them. That proprotein is released from the ribosome. It has to fold. If it's a long protein, that folding will use molecules like chaperonins that help fold it in particular ways. And then there's often also post-translational modification where particular enzymes will attach sugars, lipid components, and other things to the molecule in particular complex ways. Those tend not to be very well understood. Things like sugars are really rather poorly understood, although in the past 5 or 10 years, our understanding has gotten better. And then once it's synthesized, it has to be taken to where it needs to do its job. And there's a whole set of motor proteins that move things around to do that, which are also relatively poorly understood. Although was it last year or the year before, where the Nobel Prize was, one of the Nobel Prizes was a protein targeting. A lot's been done on it. So there are a whole bunch of steps. The idea that you just have gene and product has a lot of things between the two. And one of the key things about that is that at least 2 steps of this are progressive. Transcription, turning the DNA into RNA, requires the motor protein to actually walk along the DNA step by step and build an RNA from it, copy it one at a time, one base at a time. The ribosome walks along the mitochondrial the messenger RNA and builds the protein one component at a time, one residue at a time. And so if you have a long protein or a long gene, it takes quite a bit of time to walk through that. And there's nothing you can do really to speed that up. There's some tricks. Bacteria will have multiple RNA synthesis or RNA polymerases or DNA polymerases that walk along the same DNA at the same time. So you're synthesizing multiple copies of RNA simultaneously. But fundamentally, both of those steps take a fixed amount of time, given the length of the protein. The post-translational modification and things in the transport also takes some time. And so from the time you start transcribing a gene to the time you have any protein from that gene is typically an absolutely minimum of 5 minutes. It's typically 10 to 15 minutes. And so the cell cannot turn a gene on or off on a time scale of less than about 10 minutes, 20 minutes. And so fundamentally, that's why gene regulatory networks change the nature of the networks that are active, but they can't be used to do rapid response. If you have a cell that's going to decide, am I going to move to the left or to the right because there's a chemical I want here or I don't like there, I can't use the gene regulatory network to do it because at the time I made the decision, I'm dead. And so the reason you have these layered sets of faster, mid-range, and slower networks is because there are different timescales that are intrinsically associated with the nature of the way the networks work. So on the other hand, well, so this is just a slide going into a little bit more of the detail. Typically, when we do things like protein synthesis, we're going to assume that all the pieces that we need to make the RNA and make the DNA, I'm sorry, make the RNA, eventually make the DNA if it's a virus, DNA line, make the protein are there. We're not going to ask how much of the components do we have. We're going to assume our metabolic system's working. In reality, for example, in a cell infected with virus, you can run out of pieces and that can cause the cell to die. We also need energy to do this. So if the cell runs out of ATP, its energy currency, it's going to stop doing this thing. And there are all sorts of other very interesting molecules that do special things, like when you're copying DNA, you're untwisting a helix. As you know, if you try to untwist an old-fashioned telephone cord, it will tangle up in particularly complex ways. There are things like called topoisomerases, which run along the DNA, snip it, untwist it, and put it back together again to get rid of those kinks. And there's a whole beautiful biophysics of that we're not going to neglect. The DNA itself is not always just sitting there like this in a line ready to be transcribed. It's usually wrapped up on histones. Histones prevent transcription. And so the DNA then has to be unwound from the spools that it's sitting on to be transcribed. That winds up actually being relatively important later on when we talk about stable differentiation of cells. And then there are the whole series of other things that we care about. We're typically going to assume that all those things are there and that they work. Again, if you think about driving a car, you don't really care about how the spark plugs are firing as long as they're working. And so we're going to neglect that. But it's not that these aren't very important and interesting topics. So again, we're talking about changes of the structure of the cell in terms of its which gene circuits are on and off at a given time. There is a series of standards for describing gene regulatory networks. And the typical diagram is a line representing the DNA and an arrow coming off of it at the end to represent the messenger RNA coming off of the DNA. And what you'll see at the back end are a bunch of signals that are coming in that are determining whether that gene is being transcribed or not. And one of the problems that we're going to face when we build models is that if you have multiple signals coming in, you don't really know how to combine them. If I have two signals that both promote gene X being turned on, do I need both of them? Do they add? Do they multiply? How do they interact? If I have one signal that promotes the gene and one signal that inhibits the gene, does the promoter win? Does the inhibitor win? What happens? There's no universal rule for that. It's different from case to case. But typically what we're going to be asking is, what is the rate of production of a molecule that is a protein? And how does it differ between cell types or between species. Again, what happens to the network's behavior as a whole if I eliminate or add genes? What are the stable states of this network? Typically, differentiation of cell types is controlled by particular regulatory genes. One of the sort of dirty secrets is that those gene regulatory networks are not stable. And so there has to be some way of locking in differentiation that's not included in the network. And that's extremely poorly understood. And then it's pretty common to have, for example, drugs that try to interfere with these networks, either turn circuits on or off in particular ways. A lot of toxicity studies also involve that. Notationally, as I mentioned, we have a picture of a gene, this bent arrow coming off of it that represents messenger RNA synthesis. And then that messenger RNA is then translated into protein. Here, the arrow isn't really being used very accurately because the gene is not used up to produce the messenger RNA, and the messenger RNA isn't used up to produce the protein. But that is the standard notation. A lot of times the messenger RNA isn't written, so you just say gene goes to protein. Here I have an inducer, I, that binds to the gene, turns the transcription of the gene on, and eventually 20 minutes later we have some protein. We're going to typically assume that the rate of production of the protein depends in a continuous way on the concentration of I. In practice, the gene is either being transcribed or not. So it's Boolean. But whether the gene is being transcribed or not turns on or off relatively fast. And so we're talking about average rates of transcription and translation. Translation tends to be smoother because you have a lot more copies of the messenger RNA. For the gene, you have one copy. And so these models are neglecting a lot of these steps. We're neglecting the editing of the RNA to messenger RNA. We're neglecting the transport of the messenger RNA to the ribosome. We're neglecting the post-translational modification of the protein. So again, we're leaving out a lot of steps. Those steps happen in different places. And so we have to think about that. We'll see a lot of different notation here. I'm not going to spend, we're a little bit behind, so I'm not going to spend a lot of time on the notation. One of the things that we're going to run into notationally is that the same name is used for the gene and the protein. And conventionally, in one case, you have the name capitalized, and in the other case, you don't, but it's not always consistent. And so there's going to, when you start dealing with gene regulatory networks, you do have to do a little bit of reading to make sure you understand what the notation is. Links can represent a lot of different things because there are a lot of different kinds of regulation of, there's a lot of different places you can change the production of a protein. I can change the rate at which the DNA is transcribed into RNA. I can change whether the RNA is edited to messenger RNA. I can change the rate at which the messenger RNA is translated into protein. I can change the rate at which the protein is modified after it's been synthesized, or the rate at which it's transported to where it needs to be, or the rate at which it's broken down. And so there are a lot of steps, and typically all those steps are going to get neglected. We're just going to say molecule A increases or decreases the rate of production of protein B. Sometimes we'll put in a delay step because that time delay of 15 to 20 minutes can be really critical. You'll get a lot of oscillations in gene regulatory networks because of those delays. One of the problems that we're going to face is if you look at the circuit here, you'll notice on the back end of these genes, you'll see these regulators, and you'll see, for example, for Wnt8 there, you'll see that there are two arrows coming in on the back end of Wnt8. So there are two molecules that, in this case, promote the production of Wnt8. That diagram doesn't tell me whether I need both or whether one can substitute for the other or how they combine. The one next to it, BLIMP1 CROX, I will see that there are two molecules, two arrows that are coming in that are promoting it and one that is inhibiting it. I have three signals coming in. That arrow diagram doesn't tell me anything about how they interact. Which one wins if they're all three there. And so this kind of diagram is designed to make these circuits look simpler than they are. Again, it's not a counsel of despair. People build these models all the time and they work surprisingly well. In fact, they build Boolean models where these circuits, where the genes are either on or off, and they work surprisingly well. But you have to always remember that we're simplifying things a lot, and that simplification is something that we have to be conscious of. The other thing that happens, and I mentioned before, is that DNA is not DNA. Yeah, please. Well, so one thing that you'll find is that protein names tend to be historical. First, they tend to be created because people name them after themselves. They named them after their dog. They named them after a fruit. They named them because they thought it was cute. So we will run into proteins called frizzled, Van Gogh, flamingo, rutabaga, socks, oct. Something with, if it has an X in it, it usually turns another gene on or off as it happens. So there are a few conventions, not always, but usually. But you will find a lot of dash and a number. So you will see something like FGF, fibroblast growth factor. And then you will see FGF1, FGF2, FGF3, FGF4, FGF5, FGF6. That was because somebody discovered that there was some chemical that had the result of causing fibroblasts to grow. This was probably pre-modern genomics and proteomics. And then they discovered there actually were many chemicals that did similar things, and they gave them numbers. And then very often you will see 1A, 1B, 1C, and 1D because they discovered that one was not one thing, but there were multiple versions of it. So a classic example of that would be something called VEGF-A, vascular endothelial growth factor A, which won Judah Folkman Nobel Prize many years ago. So VEGF was discovered as vascular endothelial growth factor. And then there were a whole series that were discovered. In that case, they were given letters rather than numbers. But then it was noticed that VEGF-A, vascular endothelial growth factor A, which is one of the key regulators of blood vessel growth in tissues. It's necessary to maintain tissue integrity, but it also is involved in cancer, which is how it was discovered. It has different splice forms. So there are what are called long isoforms of VEGF-A and short isoforms of VEGF-A. Some of them have patterns that bind to specific extracellular chemicals, extracellular matrix protein. So the long isoforms of VEGF-A are secreted by the cell and they immediately glom on to the environment of the cell and they sit there. So they're basically sequestered in the environment as resources. The short isoforms don't bind and they diffuse and act as signaling molecules. And so the long isoforms tend to provide spatial information to cells, and the short isoforms act as growth factors, growth promoters. They are often indicated with subscripts that represent the molecular weight of the species. So VEGF-A 134. where 134 isn't the 134th one that was discovered, it's the one that weighs 134 Daltons, kilodaltons. It's not entirely consistent, but there is some attempt to do this. In our world, recently we've been doing a lot of things with immune response and you'll have some of that interleukin. Interleukin was discovered again before all the molecular detail was understood. Then you came up with interleukin 1, interleukin 2, interleukin 3. They're up to what, 26 or something like that now? 27, okay. And then interleukin 1, well, now it's interleukin 1 alpha and interleukin 1 beta, which are actually quite different. There's a lot of splitting and grouping. Again, from the point of view of the math we're going to do, it won't matter, but from the point of view of actually trying to read the literature, the other thing that you'll find, and Wikipedia is very good at mapping these things, it'll say, well, interleukin was discovered by so-and-so and they called it such and such, and then somebody else discovered the same molecule and called it something different, and then somebody else called it something different. And there are resources online, like I probably won't do the ontology lecture this time because it gets people's eyes glaze over when I do it. There are online resources. There's a website called Bioportals, which you type the name of a molecule and it will give you a page which says this is the official definition of the molecule. This is what the gene sequence is. These are all of the other names that are used to describe it and so on. If you're just dealing with current literature, you're probably okay. Although even today, sometimes there are different names in different fields. So the same gene in fruit flies, humans, and chickens often has a different name because they were discovered independently by different people. Sorry, Joel. Usually one name eventually wins, but there can be long periods when the usage is about 50-50, and then it can take a long time for the symmetry to break and somebody will decide. And if you're reading older papers, you're stuck. There was something funny that for IL-1 beta, there's a note in Wikipedia that a lot of people somehow use the German long S, the double S symbol, instead of beta. So that if you want to look up papers for interleukin 1 beta, you have to also look up interleukin 1 SS, because otherwise you'll miss a third of the paper. It reminds me a little bit of learning to read Japanese, where every kanji symbol has at least 2 and often 6 or 7 different pronunciations and multiple meanings. And every meaning has multiple kanji, and every pronunciation has many, many meanings and many, many kanji. There is a sort of a relationship between these things, and people can learn it, but it's certainly not a simple one-to-one relationship, which is what you'd like. So again, from the point of the mathematics of this course, this is not going to make a difference. But if you're doing research to try to say, what gene network am I going to model? Boy, it's a mess. It's a mess. And I added a homework problem this week, an open-ended one, which is I said, please try to find some resource where you can find a gene regulator, a network, and just pull a network and tell me about it, just to get a sense of what that complexity is. I was mentioning that the transcription of DNA, DNA isn't just DNA. DNA gets modified in a whole variety of ways. It can be methylated. Methyl groups can be attached to the DNA, which turn it off pretty permanently. It can get wrapped in chromatin, and depending on what is attached to the histones on that it's wrapped around, that can affect the rate of transcription. The genes could be physically remodeled in the nucleus to certain positions where they're transcribed more easily or less. And so there are a whole series of things that modulate the rate at which genes are transcribed that are not present in a diagram like this. None of those things are here. And they're all actually more important, especially to turning genes off, than the things that are here. What's known is that if a gene tends to be turned on a lot through this kind of mechanism, somehow the cell reorganizes the gene so that it's easier to transcribe it. It'll demethylate it, it'll acetylate the histone, it'll move the gene to the right place. If the gene is turned off a lot, then it will methylate the DNA to turn it off permanently. It will wrap it on a histone with methyl so that it's turned off. It'll move it to the middle so it's not transcribed. How those signals are gotten back and forth from whether the gene is turned on to what happens in those scales is not understood at all. And in particular in cancer, a lot of what happens is not a gene mutation, but problems with the regulation will be demethylation. The gene gets turned on by demethylation, it shouldn't be on. A histone will get modified in a particular way that shouldn't be. And there's some crazy cancer therapies where they'll demethylate everything, which you'd think would be lethal. The idea of these things being continuous in time is difficult because, of course, there's usually one copy of the DNA or two. And so to model that as a continuous variable between 0 and 1 is sort of strange. Model is an ordinary difference equation is straight. Instead, you have stochastic fluctuations. So there are an awful lot of things happening. And we also talked about the spatial relationships of transport. Again, we are going to typically just draw that. You'll see in the literature, P1 promotes P2. That means that P1 is some kind of transcription factor that binds to gene 1, either directly or indirectly, promotes its transcription, and then we'll assume that translation happens and we get protein P2. So that means as a reaction diagram, nothing is going to P2. And you'll notice that we totally neglected the messenger RNA. That works surprisingly well most of the time, but it doesn't have the delays. If we care about the delays, one of the things, sorry, before we do delays, one of the things that happens in proteins is that no protein lives forever. Water pretty much lives forever. Ions live forever. ATP is pretty much forever. But proteins typically have a reasonably short lifetime, an hour, 2 hours, 3 hours. And so automatically when we draw a picture like P1 promotes P2, we have to imagine that P2 is being degraded, it's decayed. And that decay rate is typically simple, but we talked about ubiquitination. There could be active regulation of decay that we may have to worry about. And so that simple picture, P1 goes to P2, turns into that. Nothing goes to P2 at a rate that depends on P1. P2 decays at a rate that depends on P2, possibly something else. And so again, this is a little bit like learning Chinese and Japanese. I could give you some general guidance, some basic rules, and then in a particular case, you may have to check to make sure that those are the right rules to use. If we care about the delays, then we usually will include the step of the gene produces the messenger RNA, the messenger RNA produces the protein. That allows some additional regulation, but the primary thing that it does is it slows things down. And that's often pretty important. And so again, I started out with something that looked very simple. P1 goes to P2, and that actually turns into the four reactions here. Nothing goes to messenger RNA at a rate that depends on P1. The messenger RNA has a lifetime of its own. The messenger RNA promotes the production of P2 through translation, and the protein itself, P2, gets broken down. And so I can't, again, one of the reasons we're going to do these sort of open-ended exercises is because I can't help you. I can show you this, but the only way to get comfortable with it is to practice it. This is the fundamental sort of grammar and vocabulary of this world. And I wish there were a more unified and simple and organized way of presenting it, but I can't change the way the literature does it. It's not that bad. Once you do it a few times, it gets to be. We'll see things called gene regulatory motifs. This is a picture of the differentiation states in sea urchin. That wouldn't be a bad thing to do a model of as a class project. We have simple things like gene activation and gene repression. As I mentioned, here we have two repressors that come in, and we have to ask the question, do they cooperate? Do they oppose each other? How do they combine? Here, in the third one, gene cascade, an activator turns on gene 1, it makes something that turns off gene 2. That's pretty common. Those are all what are called feed forward. The inputs and the input goes to the output. What gets more interesting and is typically important in gene regulation is that usually at some point the output comes back and controls the input. Often in multiple steps, not just directly. But here I have the output of my gene produces a molecule that turns the gene off. Why would I want to do that? Well, if I want to regulate the amount of the protein that I get, the best way to do that is to have the protein turn itself off. If I don't have a lot, it's produced. If I have enough, it turns itself off. If I have the reverse, if a gene produces something that promotes itself, I get what's called bistability. If I have little of the gene to begin with, the gene stays turned off. If I have a lot, it promotes itself, and it stays on. And so that's what's called the simple latch. If I then kick it from one state to another, it tends to stay in that state. You could have molecules that affect the degree to which a molecule turns things on and off. That's its regulation by small molecules. And very commonly, you connect gene regulation with signaling. That's our phosphate group, our favorite phosphate group there, which is changing whether or not a signal is active or not. Sometimes the transcription factors complex in very, very large complexes. You'll have 8, 9 transcription factors that have to come together in particular phosphorylation states or in particular orders to turn genes on and off. In those cases, life gets pretty complicated. And notationally, it's not easy to figure those things out. With signaling networks, signals tend to be continuously graded. Genes, again, often are either on or off, or at least low rate of transcription, high rate of transcription. So they're closer to Boolean. Not always, but often. The last kind of network, and I'll skip over this pretty quickly because I do want to get to the other things in the class today, is just what are called PBPK models, physiologically based pharmacokinetic models. These are models primarily of transport. So we're operating, what we've been talking about before was within a single cell or between cells. Now we're at the scale of the whole body, primarily the blood flow, but also the lymph and other fluids in the body. Transport molecular species. The blood in particular, because it's actively pumped. If you're a bird, the lymph is also actively pumped. In humans, it's pumped, but not actively. And if we want to know, for example, how is oxygen distributed in your body, we have to know what the architecture of the blood supply is, how it goes through the heart, how it goes to the lungs, which tissues use up oxygen when. Similarly, if I want to know what happens to a drug that I swallow and it goes into my gut, I have to know where it's absorbed, how it's transported through the body, how it's deposited in different tissues. in particular, the liver and the kidneys tend to be the organs that eliminate it, how it's metabolized and eliminated there. And so you'll get these transport models, typically these compartmental transport models. And in this case, the arrow represents the transport by blood of the molecule. And this is a case where you really do use amounts rather than concentrations, because you have to be concerned about volumes of tissues. Classic PBPK problems are things like, if I take a molecule, if I take a pill, what's the amount of the chemical in each tissue in my body as a function of time after I take the pill? And I guess Giuliano was working on that for Eli Lilly in the summer. That's a big part of modern pharmaceutical pharma. If I have individuals with different weights, ages, sexes, or cardiovascular disease, those transport problems, properties, and elimination problems are going to be different. So taking the same drug with the same amount, I'll get different amounts in different places. If I made the mistake of doing something like drinking alcohol or drinking grapefruit juice, that's going to change the way my liver metabolizes molecules. And so the lifetime of those molecules will be different. If I drink grapefruit juice, typically the drugs that I take will last longer and have more effect. Unfortunately, that effect is very different in different individuals, which is why most drugs will say, don't take grapefruit juice while you're taking this medicine. And that kind of regulation is actually used clinically. HIV medications will have one molecular species that goes into the liver and prevents the breakdown of the active component in the liver. And if you're unlucky enough to get COVID, as I did in July, you take Paxlovid. Paxlovid has two proteases. One of the proteases attacks the virus. The other protease prevents the liver from breaking down the first protease. Which means that when you take Paxlovid, you have to go off any other drugs you're taking pretty much, because it changes the lifetime of those drugs. And so kinds of questions would be, suppose that I have the drug in a pill and the pill dissolves at a different rate. Then it'll be absorbed higher up in my gut, lower down. The pH will be different. And so the time evolution of that drug in your body will be different. If I inject a molecule versus swallowing it, the way its patterns of time and availability will change. These things can be pretty important because most drugs, all drugs at some level are toxic. Any molecule that does something useful is also going to do things you don't want. Your ideal molecule would only go to the places you need it and wouldn't go anyplace else. People use things like, there was a headline in some aggregated today, that nanoparticles are the future of medicine. Because in principle, you can use them to target drugs a little bit. In the case of cancer molecules, which are anti-cancer drugs, chemotherapies, which are very toxic, you could sometimes target them so that they will have, for example, antibodies on their packaging that target them to the cancer and not other tissues. Antiviral like remdesivir has all sorts of complications because the active component of remdesivir isn't something that gets into cells. So you have to have what's called a prodrug that gets into the cell, and then the cell has to metabolize it into the active component. So those are all complexities of pharmacokinetic models. And of all the kinds of modeling we've talked about, probably 98% of what's done industrially is in fact this pharmacokinetic modeling, which is what we're going to talk about the least in this class. Again, the methods we're going to learn will be important to that. And the med school had, I don't know who took over Robert's class, but maybe Sarah Quinn or somebody. But it's really, it's a beautiful topic, but very, very detailed, because in that case, you really have to know exactly what happens for each molecular type of molecule in a particular situation. But if you want to make a living modeling of this kind of modeling, that's the place people do it. Okay. In this case, nodes typically represent organs and portions of organs. Node states are amounts of molecule. You have sources like the gut, or outputs like the urine or the feces. The arrows represent transport or metabolism of the molecule. And so the structurally, these actually models are pretty simple. They tend to be linear models, but the parameters are very important and they vary from individual to individual. In reality, all of these things work together. There are chemical transformations like metabolic networks, there's activation and inhibition and signaling networks. Almost everything you do to a cell changes gene expression at some level. And there's always transport. We've been talking in PBPK about transport within the body as a whole. But within the cell, I've mentioned the words active transport a bunch of times without going into the details. Molecules have to be moved around in cells, and there are all sorts of machinery to do that. Where do you find network structures? This is an old slide. This is from a couple of years ago. There isn't a single place to go for networks. KEGG used to be the go-to place. There are a bunch, and I've asked you again in the homework to try to find some resource that you find interesting, and find one network on the resource and tell me about it. And if you find one that's interesting, we'll add it to the slide. Next year, we'll have a longer list. So let's take, I'm a little behind by this one. Let's take a 5-minute break, and then let's talk about chemical reactions. And again, if the people had, there's the homework assignment here if people need it. So I'll see you back at 6:03. Does it make more sense as we hear the same thing many times? Yeah. It's really such a rich subject. There's so many ways you could look at it. It's frustrating in a sense to have to try to talk about it for an hour. Yeah. Yeah. I always think this is the, I think you've heard me talk about it as a metaphor a lot. There's a Thomas Hardy novel, Jude the Obscure. So Jude is a peasant, and somehow he decides as a child he wants to learn Latin. And he nags the one-room schoolhouse instructor in the novel. And the school instructor eventually gives him a Latin textbook. And he looks at the Latin textbook and he realizes that there isn't some magic formula to translate English to Latin. You have to learn the vocabulary, you have to learn the grammar. It's a many-to-many mapping. It's really hard. It takes years and years and years, and you still don't learn it well. And so eventually he throws the book in the river. And so I think it's not a bad metaphor for how the relationship, we draw these diagrams as if they're something simple, and yet the abstractions work pretty well, right? Your PBPK models actually do predict the concentrations of the serum concentrations pretty well. They shouldn't, but they do. These gene regulatory networks shouldn't work, and sometimes they don't. I mean, you don't want to bet your life on a gene regulatory network model. But a lot of times, if you're a fruit fly and you're doing pair rule genes, they work pretty well. So
not only we have the biological complexity, right, and then we have the human complexity of main biological components, and it's like on top of the other.
Yeah. Well, one thing I will try not to do in this class is to pretend things are simpler than they are. And I hope I don't scare people away in the sense that to acknowledge that there can be complexity doesn't mean that the models can't be explanatory. It just means that everything we do is phenomenological, and ultimately reality is the test of whether the model works or not. If you're coming from engineering or neuroscience or a field like that, then probably people are comfortable with that complexity. If you come from mathematics or physics, there tends to be this belief that there should be some ab initio absolute truth. And in biology, boy, absolute truth is in short supply.
You think you model everything as it was supposed to be, or as a relative fact it is, and then it gives you insight into something new. And that is what Brian looking for. Why not?
No, I think it's also a question of, and I did some more sort of philosophy, but the question of what is it a model can do? And in general, a mathematical model can only ever show sufficiency. You can show that if some set of processes are active, it's possible to reproduce what you saw. But it's very, very hard to show necessity. It's very hard to show that there isn't some other way you could do the same thing. And in biology, as I mentioned, Occam's razor often doesn't work. Very often there is some other thing that does the same thing. And that happens at the cell level, at the tissue level. I mean, I take your heart out, you're going to die. That's pretty. So that's what's called canalization, where there's a single mechanism that's used to do something. That simplifies things a lot, and in engineering, we tend to do that. We tend to value what's called orthogonality, which is if I change one thing, it should change only the thing that's targeted at, not other things. Since biology isn't designed, orthogonality ain't there. And so in particular, again, when you come to cancer, more than half of cancers will knock out p53, which is involved in cell apoptosis, cell death. But beyond p53 and one or two other molecule species like APC, there's no single gene product that's interfered with in more than about 3 or 5% of cancers. Because what matters is our cells moving in particular ways, our cells dividing in particular ways, our cells dying in particular ways. There are many, many different ways you could change those behaviors to do the same thing. So for a cell to become cancerous, it has to stop recognizing certain signals that say stop dividing. It has to not die when it should die. But there are many ways the cell could die, or many ways the cells can decide to divide, or many ways the cells can decide to move inappropriately. And so there isn't a single molecule that's in control of that. p53 for apoptosis is really the only one that's like that. And so you'll hear me say that all the time. Cancer is a disease of cell behavior. It's not a genetic disease. It's not a disease of molecule. And that's true a lot. And then, of course, you could come back to this whole class and just say, why are we having a whole class on molecules if the molecules aren't important? Well, of course, cells are composed of molecules. The regulation is done by molecules. And so just in the same way, we have to understand the components and how they interact. But we have to remember that that's not everything. Okay, so again, having promised you that I wouldn't lecture too much, I wound up lecturing a lot. I appreciate your patience. So let's see. So let's talk about chemical reactions. We have already done this, so I'm going to go through this pretty quickly. We'll have arrows. Typically on the left, we have substrate. On the right, we have product. We'll try to write a rate. Sometimes you'll see something like a K, a letter K sitting on an arrow. I'll try to use V for the rate. And that tells us how much A is being produced per unit time, B is being produced by unit time, but how much A is being destroyed. I'm going to skip some of these slides. These are slides from Herbert originally, Sauro, and these are done out in the textbook in some detail, talking a little bit about how you can study rates and measure them. The point here, which I think is worth mentioning, is that we're typically going to write rates as if they depend only on concentrations. But in fact, in the real world, if I change the temperature, rates change. And you'll have exponential factors that control rates of reaction. There may be catalysts, whether you're in an acidic or acid environment. If you have ions, those all change rates. And so you have to be aware of that. This kind of diagram, which assumes that somehow this molecular reaction exists outside of the world, is really a pretty harsh approximation. And very often, for example, things like there are bacteria that swim towards higher temperatures, or bacteria that swim away from higher temperatures, or bacteria that swim towards light or away from light. That phototoxic or thermotoxic behavior is mediated by the fact that different components of the bacteria, the rates of particular reactions change differently as a function of the light concentration. And that regulates whether the bacteria swim up or down in a particular direction. And so in the case of things like phototaxis and thermotaxis, if you neglect temperature dependence or light dependence rates and their differentials, you won't get that behavior. There's a basic concept of reversibility and irreversibility. Almost all molecular reactions are reversible. If you could make something, you could break it down. If you could break it down, you could put it back together. Sometimes that verges on magic. The one that I find is magic is ATP synthase, which is a pump. And just the way a motor, if you put electric current through a motor, the motor spins. If you turn by hand the shaft of the motor, it works as a generator. As a physicist, by training, I can understand how that works. ATP synthase works as a pump. If I supply it with ATP, it spins. However, if I spin it, it generates ATP. And that one I can't understand because I don't know how to get the components there to be reacted on the bottom of the molecule. That one always struck me as a bit of magic. So there's some of these irreversible reactions that are pretty weird, but they work, and people spend their whole lives studying one little component of that as biophysicists or biophysical chemists. Typically, we'll use single arrows for irreversible reactions. We'll use double-headed arrows for reversible reactions, and we'll put a separate label, V forward, V reverse, indicate the two rates, because they're always going to be different. Positive rate means we go left to right, substrate to product. Negative rate means right to left. Typically, we will actually write a reversible reaction as two separate lines. We'd write 2 ADP goes to ATP plus AMP as one reaction. And then we write ATP plus AMP goes to 2 ADP as a separate line. There's a net rate of production, which is V forward minus V reverse. That can be positive or negative. Typically, we constrained a forward reaction to be positive. So VF has to be bigger than or equal to 0. VR has to be bigger than or equal to 0. And so those are some typical conventions, which you will see violated. Chemistry conserves mass. I've talked a little bit about how things don't conserve mass in the way we write them. If something conserves mass, it has a chemistry term, which is stoichiometric. On the other hand, when we talk about synthesis or regulation and don't consider all the compounds, we're not going to have mass conservation. And so I had a little exercise, which I'm not going to do much of, but let's just together do the first 3 of these, or the first 4 maybe. And the question is a simple one, perhaps. Some of them are pretty simple, some of them are harder. Which is the example, maybe we should, instead of doing it out, we could just do it together. Somebody could speak up and answer the question. Are these reactions as written stoichiometric, that is mass conserving or not? Let's look at number 1. I have 2 molecules of H2O going back reversibly to 2H2 plus O2. Is that mass conserving or not? Yeah, that's mass conserving. Okay. So here I have S1 is the dephosphorylated form of S2. And the phosphorylation is promoted by A. Is that mass conserving or not? That's a little bit of a cheat because I've talked about phosphorylation a little bit, but we didn't go into the details. That's one where if you have more biological background, that's going to be easy. Exactly. Yeah. Good. If I had S1 plus phosphate, goes to something. Or if I had S1 plus A with phosphate, goes to S2 with the phosphate, A without the phosphate. Then I'd have A with phosphate as a double arrow like this going out. You'll see that diagram a lot. Good. Okay, what about A goes to nothing? That's a classic non-conservation. What about our favorite molecule P53, promotive of cell death, and it's promoting cell death? Is that mass conserving? Yes, who said yes? All right, does anybody agree or disagree? That one I think we could argue about. Go ahead. Well, I think you're right. I think this is an interesting question, and it illustrates what happens when you try to combine a chemical concentration with a behavior. Because this is really about how you interpret the behavior. If the cell death means that a cell is there in my model and then it's not there anymore, then the mass of the cell has suddenly disappeared. So it's not conservation. If, on the other hand, my model somehow has the cell turned into a bunch of gunk after it dies and it's mass conserved, then it is. And so fundamentally for number 4, I would say that I failed to give you enough information to answer the question. Contribution of glucose to cell growth. That's another sort of tricky one because it's a molecular species connected to a behavior. That's a good point. So the 0th order, that's reasonable. But in fact, a lot of the glucose is used to make energy and then broken down. And so only a relatively small fraction of the mass of the glucose actually goes into creating cell mass. And I mentioned at the beginning of this wonderful book of this wonderful biological physics text of Phillips. And in the chapter 1, they actually have they don't do the calculation, but they reference the calculations. There's a beautiful database called Biology by the Numbers, a bionumbers database, which says more or less what the energy conversion rate is. And you'll see this kind of thing now in ecology, where they'll say, how many liters of water does it take to make 1 pound of beef? How many kilograms of corn does it take to produce 1 kilogram of chicken? Because there's always a conversion loss. But of course, again, if I keep track of all the downstream factors, then it's mass conserving. The question is, can I keep track of it? It's usually hard. If I try to do an energy budget for, what's the total energy usage of your car? Boy, it gets to be hard to calculate it. Am I better off running my old car? Or am I better off throwing away the old car and all the energy it took to make that car and buying a more efficient car? At least in the case of the car, we have the cost. Right? The energy used to make the car can't be more than, if I take the cost of the car and I bought gasoline, I can't have taken more than that amount of gasoline to make the car because I couldn't sell the car. But that's a pretty poor upper bound on what's going on. What about protein production from a gene with a diagram like that? Okay. Yeah, I haven't said where the components of the DNA, the RNA come from. I haven't said where the components of this protein come from that are used in transcription and translation. So the answer for most of these is no, although again, I'd be happy if you did that as a homework problem and you argued yes. If you give me a good answer, I'm happy to discuss it. And so these are the kinds of questions which I think one has to run into. Herbert coming from very much a biochemistry perspective tends to assume that chemical reactions are mass conserving. When you're dealing with gene signaling and gene regulation, most of the things you write are not mass conserving. And so a lot of the features which antimony has to do mass conservation, and it's very careful about that, are of limited use to us just because most of the molecules, it will do mass conservation automatically for you, which is very nice. If I write 3A plus 7B goes to 9C plus 10D, it will keep track of all of those together, which an ODE won't do. And so that's a very powerful feature of antimony. If I'm writing chemical reactions, it's very helpful. If I'm writing regulation and signaling, I don't use that feature that much. And this was the one where was question of how would I write this to conserve mass. And we talked about this where here I've written it where I've explicitly said that the phosphate is getting transferred as an example. All right. So now let's come back and let's simulate, do a little bit of coding today. We've got a little bit to do. The two simplest things I could write as reactions would be, I'm producing A at a constant rate. So I just have a constant supply of A, and B decays at a constant rate, which is proportional to concentration of B. And so if I write nothing goes to A at a constant rate, that's equivalent to writing the dA by dt is equal to K. And that has the form that A increases linearly in time at a rate K. If B decays, dB by dt is equal to minus K times B. That's exponential decay. That's called first order kinetics. So 0th order kinetics mean my concentration, my rate is independent of concentration. First order kinetics means it's proportional to concentration. Second order kinetics would be concentration squared. So in this case, I'm creating the molecule at constant rate. The rate of decay goes up exponentially as I increase the amount of molecule. And so now I want to define something which is a very important concept, which if you're taking something like Randy Beer's class, you'll talk about a lot. Here, the mathematics gets kicked a little bit later in the textbook, like chapter 12, which is a steady state. And we say that we're in a steady state if all of the concentrations of A, B, C, D, whatever we have, are such that the rate of change of A, B, C, and D is 0. Sometimes that doesn't, the only steady state is having all of one molecule and none of the other. And we did that where we said A goes to B, goes to C, goes to D, goes to E. If that's all that happens, we wind up with, no matter where we start, we wind up with everything is E. We don't have anything else. On the other hand, when we said E goes back to turning into A, and we let the thing run, then we get a certain amount of A, a certain amount of B, a certain amount of C, a certain amount of D, a certain amount of E, depending on the rates. And so finding these steady states is something that we need to do a lot. Typically, we'd like to be able to do that analytically before we do it numerically. Antimony has something called get steady state values, which I can never get to work. Herbert, when he uses it, it works, but when I try to use it, it never works. So I tend to just run the simulation for a long time and see what it gives me. But let's just do this. Let's do this as a little exercise. Let's take our very, very simple example, and I'll give people 5 minutes or so to do this. You may actually need a piece of paper and a pencil, which is, let's take our reaction. Nothing is going to A. So we have A being produced at a constant rate, K1, and A decays at a rate K2 times A. So first of all, what are the ODEs corresponding to this reaction? We're going to have dA by dt equals something. What is it? Secondly, how many parameters do we have? And here we have to use the word parameter is a little bit of a trick because the initial concentration of the molecule counts as a parameter. And then the third thing, which is the actual problem, which is what is the steady state concentration of A? In that case, the steady state concentration of A will depend on potentially K1, K2, and possibly the initial amount of A. And then the last thing was how is A change in time? And how does that depend on K1 and K2? We'll do that numerically. I won't ask you to do that. But why don't you spend a minute just thinking about what we've done so far, write those ODEs, or that ODE, and tell me what the steady state concentration is. Remember, the rule for steady state concentration is the rate of change of A in time is 0. So you write the ODE, you set the left-hand side, dA by dt to 0, and you solve for A star, which is the value of A at which that rate is 0. In this case, it's really simple. If you haven't done it before, it may not be trivial to figure it out, but once you've done it, it's simple. Well, let's think about this together. The first thing I have is A is being produced at a constant rate. That means I have dA by dt is equal to a constant, K1. I have A decays at a rate K2 times A. So that means A is being destroyed at a rate K2 times A. I don't have anything else in the problem. So dA by dt is K1 minus K2 times A. How many parameters do I need? 3. I need to know K1, K2, and the initial concentration of A. And I'll sometimes write A sub 0 for the initial concentration. What's the steady state? I set dA by dt equal to 0. 0 equals K1 minus K2 times A. To solve that, I just move the K2 times A to the other side, and I get A star equals K1 divided by K2. Does that make sense? The amount of A I get is K1 over K2. K1 is the rate at which I put A into the system. The more K1 I get, the more A I have. K2 is the rate at which I take A out of the system. The more K2, the higher K2 is, the less A I. So it makes sense. Go ahead. That's fine. I mean, that's correct, but if you want to know what the equilibrium concentration of A is, you have to rearrange it to solve for A. In this case, because there's only one molecular species, it's easy. If I had 15 molecular species, then I have to solve simultaneously 15 equations. If they're linear, then it's a matrix operation. If they're nonlinear, then in principle, it's not analytically soluble. I have to solve it numerically. That's the kind of place where you throw it at Mathematic and it will grind at it and tell you if there's some ugly analytical solution. I wouldn't ask you to do that. In mathematical biology textbooks, they'll give you ugly analytical solutions for some examples that you run into pretty commonly. You get K1 times K2 divided by A plus B times C plus stuff, sort of ratios of things. But typically, if the reactions aren't mass action, if you have A to the 5th in something, then you're solving a fifth order equation. You can't do it analytically, so you're stuck doing it. Yeah. That's a good question. In this case, I'm changing K1 and not K2. And so the answer is that the time scale is determined by K2 and not by K1. And it's a very good question about why that is. Why would the time scale depend only on the decay rate and not on the source rate? Suppose that I started out with, suppose that K1 were 0. So I had no source. And I start out with some amount of A. Well, what happens to A? A decreases exponentially in time. The rate constant for that decrease is K2. So the lifetime of A is 1 over K2. So essentially, this system forgets its initial state over a time scale 1 over K2. So if K2 is big, it forgets rapidly. If K2 is small, it forgets more slowly. In this particular case, I can figure that out by looking at it. In a more complex system, I might not be able to figure that out by just looking at it. And that's why I'd want to be able to do a parameter scan. I'd say, in this case, I plotted this for different values of K1. And I've noticed exactly what you said, that the value changes, but the time of equilibration stays the same. Another thing that's interesting here, and again, I'm not going to do, because of time, I think we're going to skip this exercise a little bit. We could do this analytically. is what happens to the shape of this curve, which is a question I asked that we didn't talk about yet, which is what happens to the shape of this curve in time. You'll notice that the final value, A star, doesn't depend on the initial value. Right? My A star doesn't have an A0 in it. There are other situations in which it could, but in this case, it doesn't. And so that means that if I started with no A at all, started with A0 equals 0, I don't have any A to begin with. I start out with pure distilled water. I start dumping a chemical into it. The chemical concentration will go up. I now start taking that chemical out, and eventually I'll get a steady state. That would be this picture here. I start at 0, it comes up and reaches my steady state. If I start with a lot of A, more than A star, my concentration will go down. Again, you'll notice that the time scale over which this happens is independent of the initial value of A. Because again, that time scale is controlled only by K2. And so perhaps it's worth actually firing up, and I'm debating given the time how to do this best. We'll do that in a minute. We will come back and we'll write this in tellurium and we'll do this exercise. If you change K2, what you'll see is you'll change both the time scale and the value. But if I make K2 and K1 proportional to each other, so I double K2 and double K1, what will happen? If I have some value of K1 and K2, that gives me A star. And now I double K1 and K2, what happens to A star? Does A star change? No, A star stays the same. The steady state stays the same. What happens to the time of equilibration? If I make K2 bigger, does it take longer or less time to equilibrate? Less time. So if I change K1 and K2 together, I multiply K1 and K2 by a constant, then I can change the time scale over which equilibration happens without changing the value. If I change K1 without changing K2, I change the value without changing the time scale. So I have two things that I can use independently. We're talking about orthogonality. K1 and K2 independently aren't orthogonal because K2 changes both the value of A star and the equilibration. But K1 by itself changes only the value of A star. K1 divided by K2 changes the equilibration time without changing the value. And so those are actually the two independent control parameters. Okay, so let's start with this and write a little parameter scan. This was the homework problem, the last homework problem. And so if people were able to do that, this will be a review. If you weren't able to get that to work, we'll do it to go. So let's write a little parameter scan to do this. So I'd like you to fire up antimony, and I'd like you to write in antimony this reaction. Nothing goes to A at a rate K1. A goes to nothing at a rate K2 times A. And to begin with, you can set K1 equal 1, K2 equal 1. Begin with A, the initial value of A is 0. So Do people remember how to do that from last time? I'd like people to do it on your own, but if you, but I'll do it with you while we're talking. Yeah. There are a lot of different ways you could write this. I'm not. Remember the basic antimony syntax. Or not? Oh, no. So an antimony just having nothing, it doesn't have the knot. Just blank is a knot. That's a good question. I mean, you could, I mean, there's nothing that prevents you from creating a species called X, which you use to represent nothing, and then you don't keep track of it. But in fact, that will create a new variable and it will keep track of it. That's a little bit because of Herbert thinking about this as describing chemical reactions rather than regulatory reactions and other things. That's a reasonable question. There is a concept in antimony of a boundary species, which is a species with a $sign in front of it, which is forced to have a particular value. And I'm not going to use those much in this class, but there, if you're building models that have to couple to each other, those are useful because it tells you, basically tells the people using the model that that's something that comes from the outside of the model, that it's imposed on the model rather than something that's generated inside of it. But that's a good question. There should be a null placeholder which is missing. So I'll give people a minute to try that out. If I look at what I had in my notes compared to here, I guess there's a difference. When I typed it up, I made the model into a string and then loaded the string as opposed to loading the model directly. There are a few minor differences to what I typed, but let me make sure everybody catches up here. I want to make sure everybody has that. There are a certain number of things that are optional, like whether I have a semicolon at the end of every line. The order of the lines doesn't matter. Of course, I can name my reactions different names. Those don't matter. I can give a different handle to the model string or to the loaded model, but I have to be consistent. But I want to be sure everybody's thought up. Does anybody not have that typed? Everybody got it? So the next thing that we want to do is to perhaps change the initial value of A. And why don't you do that? Let's start with, I mentioned here, I start with no A and I see the amount go up. The equilibrium should be A star is K1 over K2. It should be 1, which it is. So that worked. And now I say if I start with A equal to 10, I should see it go down. Try that. Now let's do this in a more interesting way. And this is now the first real piece of antimony coding that we're going to do. As I mentioned, this model string is a model specification. It's not a program. And that model string is read by TE load A and turned into an executable. The values of A, K1, K2, and so on in the original model specification are available in whatever the handle I gave as the name of my model when I loaded it. Here it would be M dot and the variable name or the parameter name. So M dot K1 is the value of K1. M dot K2 is the value of K2. M dot A is the value of A. I can do that on an input side or on an output side. And so if I typed originally A was 0, and now I want to try A equals 10, I can type load my model here in line 8. And now I type M dot A equals 10.0. And that will replace my initial value of 0 with a value of 10. Liana. No. If people are more familiar with dictionaries, that's certainly possible. I wasn't going to do that till later because I don't know how Pythonic people are. But yes, these are. So why don't you try that? Type M dot A equals 10, and now run it, and you'll see it behaves as if my initial value of A were 10. So this is very useful because I can load my model with some default set of parameters and values. And now I can change the model without having to change the string. And that's, again, the sort of the power of this. It's nothing very fancy, but it's useful. I make initial value of A1 here. Try it. Well, if it starts at 1, it stays at 1. Does everybody get that to work? As I said, sometimes antimony is embarrassingly simple. On the other hand, I've learned the hard way that nothing that seems simple is actually as simple as it seems. It's possible to make mistakes no matter what. And you will see me make plenty of mistakes, I promise. Okay, so now we can make a parameter scan for the initial value of A. So remember the way we overwrite the value. And the first thing we could do is just by hand, let's do m dot a equals 1, result equals m dot simulate. And let's just do one thing here. Let's not make it 1, because that's a bad, but 1 won't show us. Let's start out with m dot a equals 10. And let's only simulate up to time 2, like that. And then let's copy and simulate again and see what happens. Is it clear what I'm doing? I'm taking result equals m dot simulate 0, 2 to 100, m dot plot, m simulate 0, 2 plot. What do you think is going to happen? Am I going to get the same graph twice? Try it. Do I get the same graph twice? No. Why could that be useful? The shapes of the curve look awfully the same, but look at the values on the left-hand scale. Did everybody get that to work? What happens is that the state of your system in your simulator in R is stored after you do a simulate. And so if you do a second simulate, it continues the simulation from where you ended. So it's stored. I run the first simulate, it's up to time 2. The value of A at the end is saved. And when I run the second simulate, that's the simulate that's done forward. Because that initial value of A is only read once when I do that load A. This is useful because if I want to run the simulation for a while, see what it did, do something to it, and then continue it, I don't have to reload all the values. They're there for me. On the other hand, if I want to keep changing the value of A and starting over, it's more of a problem because I have to, instead of continuing things, I want to go back to the beginning. And so there are a series of reset functions that tell me to set my values back to the ones that were specified in that initial model specification. And why are there multiple ones? Well, it might make sense that I want to change the value of A back to its initial value. But if I changed my constants K1 and K2, I don't want to screw those up. So reset will set all the variables, A, all the chemical species, A, X, whatever it is, back to their initial values as specified in the model string. But if I've changed K1, K2, things like that, they won't be affected. If I do reset all of the values in the simulation are read back to the original values. That would be equivalent essentially to reloading the model. The difference between reset all and reset to origin isn't significant here because we haven't introduced anything else. There are other things that other states you can set in antimony besides parameters and variables. And reset to origin resets all those extra things. So we're not going to be using that now. So is it clear Is it clear what the difference is? The difference is that a parameter and a variable is important here. A variable is something that lives in a rate log, like something to A, something to B. A parameter is something that only appears in a rate in a rate on the sorry, I said rate log. A variable is something that appears in a reaction, like something goes to A or A goes to something. A parameter only appears in the rate definitions. And it may look like they're not different, but K1 and K2 don't change when I run the simulation. A changes. And antimony cares a lot about that. There isn't something that will reset the parameter values independently of the variable, as far as I know. So now why don't you try, after that first plot, put m dot reset and run it again. What happens? So this time, my value of A, I set it to 10 here. When I do reset, it reads the value here in line 6, which is 0, and it starts at 0. And so if I'm going to do parameter scans, I'm typically going to use that reset a lot. And so this is the example that we just walked. And I'm not going to go through all of the examples, but we can play around with exactly what's reset, how, and when. So now I want to do a little scan where I'm going to use a loop to change the value of A and run the simulation. And one thing I have to remember, that Python, somewhat annoyingly, doesn't have a for loop built into it that would be what I'd want as a numerical for loop, because Python for loops iterate over lists. And so standard Python only has integer for loops with the range function. So I'm going to have to use the numpy a range, and I hope that's not intimidating to people. So I'm going to have to import numpy as np. And we're going to do that enough that I would recommend always importing numpy as np. Just do it. And then np.arange gives us the loop structure that we're used to in C++ or Fortran or any other language, which is minimal value, maximal value, and step size, with the caveat that it doesn't go to the final value. The top value is not reached. So if I go 0.0 to 4.0 at steps of a half, the top value will be 3.5. So why don't people try typing this and tell me what happens? So we're going to get rid of the extras here. I'll keep my reset because I want the reset to reset my values. And you'll notice I put the reset before I change the value of my parameter. Because if I reset after I change the value of the parameter, I'll just undo what I want to just did. So here I load my model. I do for A0 in np.arange. You can also use while loops and other kind of loops in Python, but this is the one we'll use the most. Colon. You have to remember now that Python indentation has to be consistent for code blocks. Everybody try that and tell me what you got. What happened? How many plots do you get? Yeah. All right. So in our last few minutes, and I may leave to you to look at some of these slides on your own. It's not worth coming back in another class to go over these sort of details, but I do want to just introduce them to you. So that puts every plot in a different window. Now, if we were using Spider instead of Jupyter Notebooks, Spider lets you tell the IDE to overlay the plots. But that's sort of a bad choice because then what you display depends on how you configured your device. You don't want to do that. So we'll use we'll use the plot array function. We've been using something called m.plot, which is the built-in plot, which is the simplest one. There's a slightly fancier one called plot array, which we call as an argument the output of our simulator. So here I called it result. I want everybody to try that. What does that do? There are a bunch of plot functions, and if you want to be fancy and use matplotlib, you can use it. But did that work for people? If you're familiar with matplotlib, it's fine. I mean, all of these plot array and plot are built on matplotlib anyway. You get the same result, doesn't help. So, oops. So there's an option in the plot called show. Show equals false tells it to not create a new plot. So now add to your plot show equals false. And you're going to ask me, how do I remember all this stuff? The answer is I don't. I look it up. I mean, the couple that you use all the time, you remember, and the ones that you don't, you go to the manual. And again, if you're familiar with matplotlib ones, and then you use that. Show is also matplotlib. So what happens if you put in show equals false? What do you get? Just show equals false. Nothing. Because I never told it to plot it. I told it to hold it the whole time. So it's not very elegant, but I could do something like this. I could just plot the last one a second time without the false. Just show equals true. I don't need, the default is true, so I could just say without the show. Not very elegant. You've got now you've got your parameters. Everybody, yeah, go ahead. Yes. I could check if I'm in my last iteration of the loop and then change the plot to false to true. There must be a more elegant way of doing it than this, but I've never figured it out. Since result is saved, overwriting it to me seems like there is a T, I think there is a T.show command that forces the, there is something that forces it to show to display the plot without redoing the plot. You'd have to, that one you'd have to look up. Did everybody get that one? Is that right? All right. What if we make it instead of changing A0, what if we change K1? Sorry, I did this wrong. What if we, instead of changing the initial value of A, we change K1? Try that. Does that work for people? So here I'm changing the rate at which I'm producing A, and you'll notice that the final value goes up. I can change K2. So now I recommend saving these things because these are things we'll build on. We'll use these simple little code snippets again and again. Not have to reinvent them. So here I'm changing value K2 instead of K1 or A. So let's come back to A. Now I said when I change the value of K1, the value of the equilibrium goes up. But they're all blue, so how do I know? So it would be nice to be able to change the colors, and there are a whole bunch of options here that I don't always remember. There are just a couple that I would remember that I want to show you. There's a sort of an ugly name, which is reset color cycle. Why? I don't know. If I put in here in my as an option, reset, and everything is camel case, color cycle, and why is it by default true rather than false? Who knows? False. Try that. What does it do? Now they're all different things. Now I might like to have a key, and there is a sort of another awkward thing, which is labels plural equals, and now I'm going to add for each one a dictionary element, which has to be a string. And you might expect that you have to do something like an append, but you don't. And so if I then add the, and this is just for creating a key, labels equals, inside my plot, I do labels equals, and then the thing that I want to label that particular iteration of my parameter with, and that has to be in the form of a string. So if I say labels equals A0, it will fail because that's a number. So I have to convert it to a string. So here I have an example, labels A0 equals plus string of A0. Okay. It's not very elegant, but again, once you've written the code once, you save it, download it, and edit it. Now, there are a variety of commands that you can use to move that key outside of the field on the left-hand side, on the right-hand side, all of that. I'm afraid you'll have to look those up. I don't remember those commands. It's hard enough to remember labels equals. It always struck me it should be label equals rather than labels equal, but anyway, there it is. And you can also, as you like, include, you can change the X, by default, this auto scales the X and the Y ranges. You can define X limits, Y limits, logarithmic axes, label the graph, all of these things. Most, as you'll see here on the output, matplotlib, So it is using matplotlib. Most matplotlib options work. A few of them are mapped in a funny way. So were people able to get that to work? Everybody able to get that to work? So that's it. So the last thing that I wanted you to do was to do what I already showed you, which is now instead of changing A, change K1, and then change K2 and see what happens. And the key thing here is that to make that change, all I have to do is go into line 11. Instead of changing the variable A, I change K1. And maybe I should change my label so that it says I'm changing K1. And now I can see what happens. There was one other thing that I talked about, which was what if I make K1 and K2 proportional to each other? So what if I say m dot K1 is equal to A0, m dot K2 is equal to 3.0 times A0. Remember, I said that if I change K1 and K2 together, it'll change the speed of equilibration, but it shouldn't change the steady state. Let's see what happens. Well, I haven't run it long enough, but it looks pretty good. What about this one here? Why is that blue one not obeying the trend? Yeah, if I produce no A to begin with, and I start out with no A, then nothing happens. If I started out with some A, then if K2 is 0, it never equilibrates. So that the 0 case is different. Right? The rate of equilibration is 1 over K2. If K2 is 0, then it's equilibrates. Okay. So that's what I wanted to get through. And I hope people can play with that a little bit. Again, I would encourage you before you stop to go to the file menu in NanoHub and download it and save it so that you have your little example for use in your homeworks and things like that. Essentially, all the homework problems, except for that open-ended one about finding a network, are going to be just building on this simple little code. And I realized that at the last problem in the homework for today, tellurium is okay. It's not like CompuCell. NanoHub tellurium is basically contemporary. Connor has the good question about that. The one thing that's not going to work in NanoHub tellurium is that the random functions don't work in NanoHub tellurium. Late in the class when we do stochastic models, that may come up.
The reason I asked was just because I was following along, but I was doing it locally, and I didn't have to do the second statement of the plot array function to get it to show. I was able to just say false within the loop, and I was trying to figure out if that was just a difference with my more recent local download or not.
Are you using Jupyter Notebooks or are you using a different IDE?
I'm using the notebook download for Tellurium, yeah.
Okay, well, it's possible that they've changed that. To me, it makes sense that if the simulation ends, it should display what it had. At some level, yes. On the other hand, it's also doing something you didn't ask it to do, which I'm not a big believer in either.
Yeah, no,
Right.
it was more of an innocent random curiosity question. Apologies. I didn't mean to derail.
No, well, I was done. I mean, no, that was a good, I didn't know that. Thank you for letting me know. So that was new. That was news to me. Thank you, Connor. I do know that there are two bugs in the nanohub tellurium. One of which is that when you do events on the same variable and you have the event trigger at the same time, it will only execute one of them, which is incorrect, and that's been fixed in the new release. And the other one of which is that the random number generators are missing in the nanohub tellurium. That's been fixed. You can generate random numbers in NumPy so that you don't, it's not a big problem, but the ones inside of tellurium, antimony don't work. But thank you. And if people catch those kind of things and they think it's a bug, report it to me, because I talk to Herbert every other week. And we talk about tellurium and fixes. So if there are bugs, we have the person who writes the code there to report it to. Getting it rolled out into NanoHub is problematic, though, so I can't promise that we'll get it updated during the lifetime of the class, but for next semester at least we get it fixed. Any questions? I know we're just we're a minute or two over. Go ahead, please. So the first question for the mini project, I think, was to ask you to look at some papers and give me a list of papers that have some kind of network model, dynamic network model in them, that you think might be interesting. And I realized we were just starting, but I had that reference list, which I shared with you. I printed out some of it, and there also are more of them. Giuliano and Jim Sluka have added to the online version some extra ones. So you could look at those papers, or you could look at some others, but people often come to this class. Somebody might be coming from neuroscience, be interested in pulse generation in neurons, which is a network problem. Or they might be interested in gene regulatory networks in fruit flies, or they might be interested in transport networks for drugs, or goodness knows, they might be interested in economic networks for trade relationships. We've had people do that, or they might be interested in infection in influenza. And so the idea was just to find a couple of papers, maybe five papers, or it could be a Wikipedia article, something that has networks in it that might, based on the little we've covered so far, might be relevant to the class. Give us a list, write a few words, a sentence or two about why you think it's interesting, so that you could talk to classmates and say, hey, if we're going to look at this paper as a project, then the following week or the week after that, you could present it together, and we could try to narrow down what would be a good project. In the past, there are times when I've just said, you do this project, you do this project, you do this project. And if people really don't care what to do, then we could do that. But in general, people come to this class for some reason. And so I want to give you the chance of finding something that's interesting to you. So the assignment is to find 5 papers that are of interest, that have networks in them, and something that you might want to work on. Doesn't have to be one that you pick later, but of course, if it happens to be, you've saved yourself some time. And again, I realize it's tricky because until we get a little bit further along, if you haven't worked with networks, then it's a little bit hard to say, I like this. On the other hand, if I wait until halfway to the semester to have you start picking a project, I guarantee you can't finish. And so I have to push you a little bit to try to think about what might appeal to you now. And it's fine. In other words, if you come up with 5 papers and I look at it and I say, well, gee, this is what is about evolutionary biology. We can't do it in antimony. Or this is a spatial model that doesn't really work, or something else, then we can work together to try to find something that's mutually agreeable and interesting to everybody. So the goal here is not to, I understand that it seems early, but in my experience, if we don't start thinking about it and exploring, there isn't really time to, the semester's short. And we have to teach them. Basically, you're thinking about it along the analogy of learning a language. If you're learning a new language, you spend a year or two learning grammar and vocabulary before you start writing essays. In this class, you have to learn the grammar, the vocabulary, and write an essay at the end of the class. And that's a lot to do in a semester. And so we have to start, exploring what topic you might want for your essay, even if you haven't learned the language completely. And so if the answer is you don't care, and you'll partner with somebody else and you'll do what they like, that's okay too. But I still would like people to look around. Again, we've got, I think, a list of about 30 papers in that list so far. It's a little heavy on the infectious disease and cancer side of things. It would be good to have more gene regulatory networks and metabolic networks. Yes, yeah. Giuliano, could I ask you to resend that, reshare that? Sure, you can do that. It should be available to you through the Google Doc. Whereas if you go to the Google Doc, you have read access to everything in that Google, not in Google Doc, the Google Drive. You've got a link to a student materials drive folder. That. Oh, because it's a Word document. So download it and then open it. I think it's a Word doc. All right. Well, does somebody else have problems? That one, again, thank you for letting me know. We'll check it out. The other thing that's important, as I said at the beginning of the class, next week is online only because I'm traveling. So I mean, if people really want to be here and watch this on the screen here, you're welcome. I could arrange for somebody to come in and display it, but I'd guess that you'd rather not have to trek over to Luddy if you don't have to. And whether we do everything online later on or not, I'm happy to come here and lecture. If it's easier for people to stay home and watch this online, it's fine too. I think there's some advantages to be able to see you. I also know it's a pain to get here, and you may be more comfortable at home on your local computer. You may have 2 monitors and things like that. So we can revisit it. There are going to be a couple of weeks this semester when I'm traveling. And so definitively those will be online. But I hope that I'm happy to come in if people want to. You can email me privately. 1 or 2 people have emailed me privately about, I feel strongly I'd like it to be online, or I feel strongly I'd like it to be in person. You can let me know privately, not publicly. I'm open to that. So Giuliano, did you check the permissions on that?
Yeah, I'm gonna upload it.
Would that be better to have? Oh, okay. Yeah. Okay. Good. Okay, I'm sorry about that. Anybody else have any issues before we break? Okay. And again, I understand this concern about the project. If you start out by just pulling, pull 2 or 3 papers or 2 papers from that list of papers and look at them. Look at what they look like. You don't have to read them in detail. I'm not expecting people for next week. I'm not expecting people to read every word of every paper. In fact, I gave you one of the documents I gave you was a document by an old student of mine who's now a professor in Atlanta about how to read a paper. And sort of it's a little bit how to skim a paper. And so look at the abstract. Does it sound interesting? Look at some figures. Do they look interesting? Look at if there's a diagram of a network, does it look interesting? If the answer to all three of those is no, you don't have to do any more work. Throw it out and try something else. If the answer is yes, you might want to dig a little bit more. But over the years, I don't think I've ever had anybody who didn't come up with an interesting problem to work on for this class. But I don't like to prejudge it, because people have worked on all sorts of different things: immune response, influenza infection, COVID infection, cancer metabolism, developmental biology, many things. Because this network concept is so powerful. The one thing that I do want to watch out for, and another reason why I really want people to have at least a tentative project in week 4, is that occasionally people pick things that sound fascinating but have booby traps in. And the last thing that I want people to happen, and it's happened occasionally, is for somebody to be, they're determined they want to work on X. I said, well, X is really pretty challenging to do in a semester. And so I won't ever tell you you can't do what you want to do. But I will warn you if I think that it's something that may not be practical. Because I'd much rather have people do something small and finish it successfully, and then have the technique so that you can go on and do something bigger, than to try to do something too ambitious and then get frustrated. And so that's the other reason I want to get people to try to define projects early enough, so I can meet with you individually or in small groups and look at them and walk through it and make sure that there isn't anything hiding there that makes it so that in week 14 you say, yikes, this isn't going to work. Last thing I want to have happen to you is have that happen. I remember at one point somebody wanted to do a very complicated flux balance model of metabolism. And I said, well, it's a little bit, 500 rate law model is a little ambitious. At the end, it didn't work. And I think they learned something from doing it, but still, it's a lot more satisfying if at the end of the semester you can present a project that shows everybody what happened, and it works, and you can upload it to NanoHub. And you could say to some future employer or graduate school or something else, this is something I did, you can see my work. So my goal here is for everybody to learn and succeed. And so I'm not trying to be a hard ass about getting things done early. But again, the reason I'm doing it this way is to try to make sure that we set everybody up to succeed. Any other comments or questions? We're a little bit over, but I hope that was okay. Connor, I know you've been breaking in when you needed to, so I assume if you had something to say, you would say it.
Yeah,
Okay.
absolutely.
Okay, thank you. Good night, everybody.